It is my pleasure to introduce you to this commentary published on iMeta. The title is Role of Respiratory System Microbiota in Development of Lung Cancer and Clinical Application. Microbes play a significant role in human tumor development and profoundly impact treatment efficacy, particularly in immunotherapy. The respiratory tract extensively interacts with the external environment and possesses a mucosal immune system. This prompts consideration of the relationship between respiratory microbiota and lung cancer. Advancements in culture-independent techniques have revealed unique communities within the lower respiratory tract. Here we provide an overview of the respiratory microbiota composition, dysbiosis characteristics in lung cancer patients, and microbiota profiles within lung cancer. We delve into how the lung microbiota contributes to lung cancer onset and progression through direct functions, sustained immune activation, and immunosuppressive mechanisms. Furthermore, we emphasize the clinical utility of respiratory microbiota in prognosis and treatment optimization for lung cancer. Niche-specific bacterial communities inhabit the surface of the adult respiratory tract with a roughly decreasing trend in bacterial burden from the upper to lower respiratory tract. The microbial communities in the lower respiratory tract and lung are predominantly composed of bacteria belonging to the genera including Prevotella, Streptococcus, Veillonella. This microbial composition undergoes certain changes in lung cancer patients, as shown in the figure below. Additionally, recent research has progressively revealed the unique microbiota within lung cancer tissues, which has been found to be closely associated with the biological behavior of lung cancer. It is also crucial to acknowledge that respiratory bacteria are significantly influenced by environmental factors and individual physiological states, such as seasons, climate, pollution, age, smoking, gender, antibiotic usage, and even pet ownership. The lung microbiota has been demonstrated to contribute to lung cancer development primarily through direct biological functions, sustained immune activation, and immunosuppressive mechanisms. Microbial dysbiosis can directly induce genomic damage and instability through reactive oxygen species or genotoxins. DNA damage within respiratory epithelial cells can progressively accumulate and eventually trigger carcinogenesis. Additionally, some microbiota can induce the upregulation of specific signaling pathways, influencing the growth, apoptosis, and proliferation of respiratory epithelial cells. The local metabolite network can also be influenced by bacteria, with specific metabolites potentially participating in tumor cell metabolic processes, thereby promoting tumor development. On the other hand, bacteria can indirectly promote tumor progression through interactions with the respiratory mucosal immune system. Initiate procarcinogenic inflammation via toll-like receptors. Microbiota in the lower respiratory tract and tumor tissue can recruit immune cells closely associated with tumor progression, such as T helper 17 cells and neutrophils, thereby promoting tumor advancement. Interleukin 17, interleukin 6, and other pro-inflammatory cytokines also play a crucial role in this process. In response to excessive activation of the innate immunity, epithelial cells may initiate transcriptional programs to dampen innate signaling, and regulatory T cells and M2 macrophages are induced to limit inflammation. These changes may allow lung epithelial cells, which have already accumulated tumor characteristics, to evade immune surveillance and transform into tumor cells. Bacteria also participate in this process. It has been demonstrated that some bacteria can expand the pool of immune cells, such as regulatory T cells and M2 macrophages, by influencing the expression levels of specific genes. In summary, advances in culture-independent techniques have recently revealed previously unrecognized microbiota in the lower respiratory tract and lung cancer. Furthermore, lung or intratumor microbiota can contribute to lung cancer progression through direct biological functions, sustained immune activation, and immunosuppressive mechanisms. Dysbiosis of the lower respiratory tract microbiota can serve as a potential therapeutic target. However, its clinical value warrants further investigation. The safety of aerosolized bacteria should be also carefully evaluated.